This is the start of the Unit 1 lecture, and I do want to point out that before you start this video, I would prefer that you go into Moodle and watch this YouTube video first before you watch the lecture video. It just gives you kind of an overview of some of the immune cell function basics, and you can see it's a quick video. It's only 6 minutes, so watch that one first. It takes you to Professor Dave. We've got a few of his videos throughout this semester. He does a pretty good job of explaining some stuff, but again, that just gives you an overview of what we're going to talk about in this lecture. Okay, so for the lecture, this first little bit is going to be on the topic of immunity. And we kind of hit on some of this in micro last semester. But we're going to get, and you guys, touched on blood bank in intro, but we're going to get much, much more in depth into a lot of these topics this semester. And blood bank, I think you guys will find maybe a little bit more relatable than micro, but it is some very complex topics. So just a lot to go through here. Okay, so like I said, this first lecture is on immunity. So a little bit of terminology. So immunology is defined as the study of a host's reaction to foreign substances that are introduced into the body. And usually that is introduction of a foreign antigen. So this is a foreign substance that induces an immune response in the host. So we have our own antigens on our own cells. So it's the fact that you're being introduced to a foreign antigen that induces that immune response. So, and we had talked a little bit previously about autoimmune disorders. So that's kind of a default or a defect in the mechanism of your body being able to recognize something as self versus non-self. So what happens is in autoimmune disorders is the body sees its own cells but thinks that they are foreign. So it induces that immune response. And then immunity is either innate, which is also referred to as specific, or adaptive, which is, this is supposed to be non-specific, or adaptive, which is specific. And we talked about that a little bit previously also. So the individual's ability to resist infection by means of normally present body functions. So that's just your, we talked a lot last semester in micro about host defenses. So that's what this is referring to. So those natural, non-specific responses that occur in your body when it is introduced to a foreign substance or a foreign antigen, it's going to respond the same way no matter what that foreign antigen is. So it's non-adaptive, non-specific. So the response is going to be the same for all pathogens or foreign substances that it encounters. So no prior exposure is required. And because of that, that immune response lacks any kind of memory and specificity, but the effect is immediate. It recognizes that something foreign is there that's not supposed to be there, and your body immediately reacts to start fighting off that foreign invader. So those mechanisms or your body's ability to respond is influenced by nutrition, your age, fatigue, how much stress you're under, and then there is also a genetic component as well. Okay, so some of those external defenses composed of physical, chemical, and biological barriers that function together to prevent most infectious agents from entering the body. So we talked about all of these last semester in micro. Your skin, it serves as that initial structural barrier to prevent and protect. Secretions on the skin discourage the growth of microorganisms. Your mucosal membrane surfaces, they're going to block adherence of bacteria to the epithelial cells, because remember, they kind of hold on to the bacteria to prevent them from going further into your body. coughing, and sneezing help move pathogens out of the respiratory tract. So they're going to expel any trapped bacteria or foreign pathogens. Sweat, the lactic acid in your sweat helps control the pH of the skin to help prevent the growth of bacteria. Your sebaceous glands include fatty acid secretions that also help to control pH, that help prevent the growth of the bacteria. Stomach acid, so remember that hydrochloric acid that's produced in your stomach has a very, very, very acidic pH, a very acidic environment. So a lot of bacteria are not able to survive in that environment. Cilia, so remember those are those kind of like little hair-like structures. Motion of the cilia in the nasopharyngeal passages and in other places in the body clears almost 90% of the deposited materials. Tears and saliva produce the enzyme lysozyme that attacks the cell wall of different bacteria, especially the gram-positives. Your normal flora, otherwise known as your microbiota, help fight off pathogenic bacteria because remember they're competing for the same environment and the same nutrients. So your normal flora wants to maintain its hold, so it's trying to fight off those bacteria. And the genitourinary tract, so the flushing action of the urine and the acidic pH of the vagina can help control pathogens. Okay, and then with innate immunity, so if the microorganisms or that foreign pathogen is able to get beyond the external defense system, so we've got additional defenses that are in place to help mitigate that. So the cells of the innate immune system, so we've got white blood cells, specifically we've got phagocytes, which are, sorry, I skipped over a slide there, which are, that translates to cells that eat cells. And if you remember from last semester, I referred to them a lot of times as like the little Pac-Man cells because I just imagine them going throughout the body and gobbling up all the foreign antigens. So they help with the destruction of foreign particles. So the main purpose of the inflammatory response is to attract cells to the site of the infection or the foreign particle and remove those foreign cells or pathogens through this process of phagocytosis. So the structure that results in response to that microorganism being engulfed by the cell is referred to as a phagosome. So the phagocyte ingests the foreign pathogen, and that resulting structure is called a phagosome. You also have acute phase reactants that enhance this process, but your cellular elements are going to have the biggest role. So the following cells are the most active in phagocytosis. So your neutrophils, they are the ones that are going to play the primary role in phagocytosis. They make up about 50 to 70% of the total white cells in an adult, in a normal adult. They increase in circulation almost immediately in cases of acute infection, and they're going to go rapidly to that site of infection. Then we have eosinophils that make up about 1 to 4% of the total circulating white cells in a non-allergic person. Numbers are going to increase in allergic reactions or parasitic infections. So if you're a person who constantly is fighting with allergies, seasonal allergies, your eosinophils might be increased on your CBC. The most important role for the eosinophils is in cytokine release. Cytokines are chemical messengers in that adaptive immune response. Okay, then we have basophils that make up less than 1% of all of your circulating white cells. They're the smallest of the granulocytes. They can induce and maintain allergic reactions. They can help with regulation of some T helper cell responses and stimulate B cells to produce an immunoglobulin known as IgE. And then your monocytes that make up 2 to 10% of your total white cell population. will go on to become macrophages. Okay, then our tissue cells, that macrophage, so this is a type of tissue cell that comes from the monocyte, and they're going to have specific names depending on the location that they originate from. These macrophages play an important role in innate and adaptive immunity. The killing activity is enhanced when they become quote unquote activated by contact with those foreign microorganisms or with those cytokines that are released by certain types of lymphocytes during the immune response. So remember, we said just a second ago that the cytokines are chemical messengers. So lymphocytes come in contact with this foreign particle. They release these cytokines that are the chemical messengers, and it's kind of like it's sending out messages throughout the body that something's wrong and we need to take care of it. They have a major role in the adaptive response by presenting those foreign antigens to our T lymphocytes. Okay, and I said that they, it depends on, the name of the cell depends on where they originate in the body. So alveolar are in the lungs, Kupffer cells are in the liver, microglial cells are in the brain, histiocytes are in the connective tissue, and osteoclasts are in the bone. Okay, then our mast cells, they resemble basophils, but they're from a different lineage. They're found in skin, connective tissue, mucosal tissue of the respiratory tract, genitourinary tract, and digestive tracts. They're going to increase vascular permeability and increase blood flow to the affected area. So increasing vascular permeability, increasing blood flow just means that that reaction time is going to be faster to react to that foreign pathogen. Mast cells play a role in allergic reactions, but they can also function as antigen presenting cells, or APCs. So again, our body is recognizing that foreign antigen, which is just like a marker on the cell. You can think of it as kind of like as a little flag that's standing up on the surface of that foreign cell. So it recognizes that foreign antigen and it can take that cell and present it. Dendritic cells are also classified according to their tissue location. So after capturing an antigen in the tissue through that process of phagocytosis or endocytosis, they travel to the nearest lymph node and present this antigen to the T lymphocytes to initiate that adaptive immune response. So dendritic cells are considered the most effective antigen presenting cell in the body, as well as the most potent phagocytic cell. Okay, pattern recognition receptors. So macrophages and dendritic cells they play the most important role in pathogen recognition. They're also found on neutrophils, eosinophils, monocytes, mast cells, and epithelial cells. So you've got these receptors on these cells that bind to a pathogen. Phagocytic cells become activated and they're better able to engulf and eliminate those foreign microorganisms. Cytokines and chemokines get secreted. So this is, again, chemical messengers. It's recognized that this immune process is occurring. We're sending out these chemical messengers or these signals to our other cells. So this is what triggers the adaptive immune response. Those pattern recognition receptors also are able to distinguish self from non-self by recognizing substances known as pathogen-associated molecular patterns, which are only found in microorganisms. So examples of that would be like the peptidoglycan in your gram-positive cell, in the cell wall of your gram-positive bacteria, lipopolysaccharides in the gram-negative bacteria, zymosan in yeast, and flagellin in bacteria that have flagella. And then our toll-like receptors, so this is a family of receptors located in various cellular compartments. Each of them is made to recognize a different microbial product. And then once it gets bound to that substance, that the host immune response is activated. Okay, acute phase reactants. So these are normal serum constituents that act as a non-specific indicator of inflammation. So these acute phase reactants are going to rapidly increase in the blood or decrease in concentration due to infection, injury, or trauma to the tissues. And I'm sure you guys will talk about these a lot more in chemistry this semester. A lot of these proteins act by binding to microorganisms and promoting adherence, which is the first step in phagocytosis. Other acute phase reactants help to limit destruction caused by the release of proteolytic enzymes from white cells as the process of phagocytosis takes place. Some of the most important acute phase reactants and some of the ones that we most commonly measure in the lab are C-reactive protein, or CRP, which is synthesized in the liver. It's like an antibody because it is capable of opsonization, which that term means the coating of foreign particles with antibody. It's also capable of agglutination, precipitation, and activation of complement by the classical pathway. We're going to talk a lot more about complement activation coming up. However, binding is calcium dependent and non-specific. You'll see elevated levels of CRP in bacterial infections, rheumatic fever, viral infections, malignant diseases, tuberculosis, and after a heart attack. Because the levels rise and then decline so rapidly, CRP is the most widely used indicator of acute inflammation, but it's a non-specific indicator. So you can have an elevated CRP, and again, it's a commonly ordered test because it does indicate infection or inflammation, but it's non-specific. So just because your CRP is elevated, the doctor may not know what's causing that. Serum amyloid A, also synthesized in the liver, appears to act as a chemical messenger, so it's kind of like a cytokine, and it activates monocytes and macrophages to produce products that increase inflammation. It's going to be this serum amyloid A level is going to be increased significantly more in bacterial infections than in viral infections. So if and serum amyloid A is not as common of a test as the CRP. A lot of times those are going to be send out tests. So, but it's a little more specific as to the origin of the inflammation or infection. Alpha-1 antitrypsin, synthesized in the liver, it counteracts the effects of neutrophil invasion of the lungs during an inflammatory response. So if there's some type of pathogen invasion that's going on in the lungs, and this whole immune response has been activated, and you've got neutrophils that are going to the site, which is the lungs. This alpha-1 antitrypsin can help reverse or kind of mitigate or at least control some of that inflammatory response, because it's hard on your body. Like, your body's doing what it's supposed to do. It's fighting off that infection, but at the same time it's releasing all these like danger chemicals. So it does affect your body. Haptoglobin binds to free hemoglobin released by intravascular hemolysis. So it acts as an antioxidant to provide protection against oxidative damage caused by that free hemoglobin. So intravascular hemolysis, your red cells are lysing. It causes free hemoglobin to be released, so this kind of protects against that. The haptoglobin will bind to it. Increases in haptoglobin levels can be seen following inflammation, stress, tissue necrosis, and levels of haptoglobin may initially decrease because of intravascular hemolysis, but then they'll increase. Fibrinogen is an acute phase reactant involved in the coagulation pathway. So maybe you guys did talk about that last semester. Forms a clot that increases the strength of a wound, and it stimulates endothelial cell adhesion and proliferation, which is critical to the healing process. So the formation of a clot creates a barrier that helps prevent the spread of microorganisms further into the body. And then, excuse me, fibrinogen makes blood more viscous and promotes aggregation of red cells and platelets. So all of this just kind of helps that clotting process, kind of helps protect the body against the foreign pathogen. Ceruloplasmin is the principal copper transporting protein in human plasma. So copper is normally absorbed out of circulation by the liver and either returned to the plasma or excreted in the bile duct. There's a condition called Wilson's disease where copper accumulates in the liver and in other tissues such as the brain, corneas, kidneys, and the bones. Okay, inflammation. So that term it just refers to the body's overall reaction to injury or invasion by an infectious agent. So the 4 cardinal signs or clinical symptoms of inflammation are redness, also referred to as erythema, swelling, which is referred to as edema, heat, and pain. And I know anytime you've had, like a scratch or some kind of wound, you've noticed that. Even just from like maybe like a bee sting, it'll get red, it'll swell, it'll be hot to the touch, and it might hurt. Complement components. So these are serum proteins that are normally present in an inactive state that contribute to inflammation. So once they're activated, zymogens, which are precursors of enzymes, they get converted to active enzymes that enhance that immunologic process. They're heat labile. Most are going to be synthesized in the liver except for C1. The major functions of complement are opsonization, chemotaxis, and cell lysis. There's also several regulatory proteins that are involved in this process that help disrupt the complement activation process in a regulatory fashion. So again, your body's doing what it's supposed to be doing, but at some point it has to, there has to be like regulation in activated also to help prevent your body from over responding. So some of those regulatory proteins are factors H, I, S, and B. So there's excess activation in septicemia, tissue necrosis during myocardial infarction. So in order to prevent that excess activation, we have to use these regulatory proteins. So deficiency in these regulatory proteins leads to increased susceptibility to infection and a delay in clearing those immune complexes. And the classical pathway, so complement activation can occur in a few different ways, either through the classical pathway, the alternate pathway, or the lectin pathway. So the first one, the classical pathway, you've got 9 complement proteins that get activated by bound antibodies. So, and remember, our antibodies are what are produced in response to a foreign invader. We do have some naturally occurring antibodies. So they're initiated by those antigen antibody complexes. So the binding of the antigen with the antibody. It needs calcium and magnesium for activation. This only includes your IgG or IgM antibodies. If it's IgG, it requires 2 for activation. If it's IgM, only 1 antibody is required because the IgM molecule is larger. So 2 IgG or 1 IgM antibodies can bind complement. Okay, the recognition unit of the antibody recognizes the Fc region of the antibody. We're going to, and this might seem a little bit foreign to you right now, but we're going to look at a diagram of an antibody in just a minute. Calcium is required, so this whole process is held together by calcium. You've got chelating agents that can destroy C1 because they bind calcium, such as EDTA. And chelating, I don't remember if you guys would have talked about this previously, probably in hematology, but chelating just means that it's an agent that is used to bind to another agent. Then you've got C4, C2, and C3, which are the activation units. So C4 is the first in the amplification step. C3 is a major component and second amplification, and then complement components C5 through C9. These are referred to as this membrane attack unit. So it's initiated with the binding of C5. And I know this sounds kind of weird and confusing. There is a video in Moodle that has a lot of diagrams. The guy does a really good job of kind of going through the different things that are happening. So I would recommend taking a look at that. He'll go through each one of these. I think he goes through all three: the classical, alternative, and the lectin pathway with diagrams and just kind of explaining what's happening. And here is a very simplified version of the complement cascade in each one of these pathways: classical, the lectin, and alternate pathway, and then what's happening through each of these. I think I got off track here. Okay, so that was classical. Okay, so the alternate pathway of complement activation. So microorganisms can trigger additional complement numbers in an alternative pathway. So this one is antibody independent. So we said that the classical pathway was dependent on that antibody antigen complex once they bind together. So this one is the alternative pathway is antibody independent, which means it's not dependent on that antibody. It's triggered when C3b directly binds to a microbe, and that can be bacterial or fungal. Could also be foreign materials and damaged tissues. So the first step is at C3, where factor B binds to C3, then factor D binds. Then factor B, D, and properdin are required. The alternative pathway, C3 convertase, can cleave or break apart multiple C3 proteins into C3a and C3b, which is unstable until it binds that properdin, which is a serum protein. Then you've got C5 convertase from either pathway, follows the same path regardless of how it's activated. And then C5 convertase cleaves or breaks apart C5 into C5a and C5b. C5b binds sequentially to C6, C7, C8, and then to multiple molecules of C9 to form a membrane attack complex. So those regulators, regulatory molecules, most work at C3. Factor S is a control protein and a regulator at the C5 complex. C5a is a potent anaphylotoxin and chemotaxin that functions to increase vascular permeability and attracts white cells to the site of the inflammation. And factor H is an alternative pathway regulator. Okay, and then finally, your lectin pathway represents another means of activating complement. So it's activated by direct recognition of surface molecules. So this is just this pathway is an additional link between the innate and acquired immune response because it involves nonspecific recognition of carbohydrates that are common in microbial cell walls. It plays an important role of defense in infants in the period between their loss of maternal antibodies and the development of their own antibodies. Early lectin molecules are structurally similar to those in the classical pathway. So your recognition molecules in the lectin pathway are lectins, phycolins, and collectins. The mannose binding lectin pathway, which it's also referred to as, is or mannose binding lectin is a key protein that binds to mannose or other related sugars in a calcium dependent manner that initiates the pathway. Those sugars are found in the microorganisms. Mannose binding lectin is an acute phase protein produced in the liver that increases during initial inflammatory responses. And then after the mannose binding lectin binds to mannose, serine proteases that are referred to as mannose binding lectin associated serine proteases, which are associated with this complex. So after that point, activation of the lectin and classical pathways is the same. Okay, so to have said all that, I know it sounds very confusing and you've got all these C1s and C2s and B and Ds, and I get it's confusing. Watch that video in Moodle. But really, if you just kind of pay attention to this little flow chart here, that I think that just gives a visual to make it a little bit more relatable. But the biggest takeaways here are what activates each one of these pathways. So the classical is antigen antibody complexes. Lectin is, alternative was the second one we talked about. It reacts to actual pathogen, the surfaces of pathogens. And then the lectin pathway is kind of a little bit different. Mannose binding lectin or phycolin binds carbohydrates on pathogen surfaces. Okay, so and now would be a good time. There is that video in Moodle on complement pathways. Now would be a good time maybe to pause and watch that video. Okay, some different organs in the immune system. The bone marrow and thymus are considered the primary lymphoid organs where maturation of your B lymphocytes and T lymphocytes takes place, respectively. So the B lymphocytes in the bone marrow, T lymphocytes in the thymus. The secondary lymphoid organs provide a location where contact with those foreign antigens can occur. So your secondary lymphoid organs include the spleen, lymph nodes, appendix, and other different types of mucosal associated lymphoid tissues or MALT. The primary and secondary organs are differentiated according to their function in adaptive and innate immunity. So your primary lymphoid organs, again, your bone marrow, which is involved in antigen independent lymphopoiesis, and B cell maturation and differentiation takes place here. And then the thymus, which is located in the chest cavity below your thyroid gland, it's the largest at puberty, then it's going to shrink after that. The majority of the T cells that your body produces are produced early in life, and then there's going to be a reduced rate later in life. T cell differentiation occurs early in fetal development, and then maturation of the T cells over 3 weeks. Different surface antigens are expressed as the cells mature to protect against different invaders. Mature lymphs are then released from the thymus. Okay, your secondary lymphoid organs. So once the lymphs mature in those primary organs, they're released to the secondary lymphoid organs, which again, that includes the spleen, lymph nodes, cutaneous associated lymphoid tissue, abbreviated as CALT, and then your MALT that was mentioned just a minute ago in the respiratory, GI, and urogenital tracts. So you've got constant recirculation of lymphocytes through the organs to the bloodstream. So that allows for contact if there are foreign antigens present. That constant circulation is going to allow for the body to more quickly and better recognize those foreign antigens. Lymphopoiesis, which is your multiplication of lymphs, occurs in the secondary lymph tissue and is dependent on antigenic stimulation. So again, your body has to respond to a foreign antigen. The spleen is the largest secondary organ. It filters old, damaged cells and foreign antigens from blood. It's made up of red pulp and white pulp. So the red pulp makes up a little over half of the total volume. It's rich in macrophages. It helps destroy old red cells, platelets, and some pathogens. And then white pulp is about 20% of the total weight of the spleen, contains the lymphoid tissue arranged around arterioles in a periarteriolar lymphoid sheath. And then that sheath has a lot of T cells. Attached to the sheath are primary follicles, which contain your virgin B cells, which just means that they have not yet been stimulated by those foreign antigens. And then the lymph nodes are located along the lymphatic ducts and are especially numerous near the joints and where the arms and legs join the body. So they're kind of central collecting points for lymph fluid from those adjacent tissues. The lymph fluid is just a filtrate from your blood vessels. Its function is filtration. So fluid moves through the sinuses lined with those macrophages. So this is a site of phagocytosis and antigen processing, and also generation of your B cell memory occurs here. So the secondary follicles, you've got antigen stimulated proliferating B cells. T cells are in the area next to the antigen presenting cells. If antigen contact takes place, which means our cells have come in contact with a foreign antigen on a foreign particle, lymph movement stops for 24 hours. And the reason, so you've got this lymph fluid that's moving, it's constantly in circulation, it's looking for foreign particles. If it comes in contact with one of those foreign antigens, the lymph fluid halts, it stops. And the reason for that is so that it can immobilize the antigen. When that happens, your lymph nodes will enlarge. Maybe you've noticed that happening before. Okay, the MALT, additional areas of lymphoid tissue include the mucosal associated tissue, known as MALT. So this is found in the gastrointestinal, respiratory, and urogenital tracts. Some examples include the tonsils, your appendix, Peyer's patches, specialized type of MALT that's located at the lower ileum of the intestinal tract. So these surfaces represent some of the main ports of entry for foreign antigens. You've got numerous macrophages and lymphs are localized here so that they can recognize and start to respond to those foreign antigens. And then CALT, or your cutaneous associated lymphoid tissue. So skin is considered the largest organ in the body, and epidermis contains several intraepidermal lymphocytes. Most are going to be T cells that are positioned to combat pathogens that enter through the skin. Okay, so all of that, so far we've been talking about innate or non-specific immunity. So now we're going to talk about adaptive or specific immunity. So this type of immunity is specific for each pathogen or microbial agent. So the body can remember previous exposures. Because of that, it results in an increased response. So the response time can be longer, but it's more pathogen specific. So remember when we were talking about innate immunity, this is just your non-specific natural host defenses that are activated with any kind of foreign pathogen, and it's immediate response. So this is kind of the opposite. Our body has already been in contact with a pathogen previously. It has learned that pathogen, and so now if it's exposed to it again, it can respond much more specifically. So this is pathogen specific, highly effective, often lifelong. So once you've produced these antibodies that are made to respond to a specific pathogen, those antibodies are usually there for the rest of your life. So adaptive traditionally has been divided into two main branches, where you've got humoral immunity and cell-mediated immunity. So your humoral immunity is mediated by soluble factors. So those are your antibodies in the liquid portion of the blood. And then cell-mediated immunity is carried out by the cells of the immune system. So you've got your T cells. The three biggest roles of the T cells are to release cytokines, those chemical messengers, to recognize self from non-self, and then they help B cells. T cells get divided into two broad categories. So you've got cytotoxic T cells and helper T cells. So your cytotoxic kill target cells that have been infected with intracellular pathogens. So that could be viruses or bacteria. And they also help kill cancerous cells. Your helper T cells, they don't kill the cells, but they control the immune response through secretion of cytokines. Again, those are just signaling molecules that allow communication between the immune cells. The T helper cells they get further divided into several different subpopulations based on the types of cytokines that they secrete. Okay. T cell differentiation. So soon after the T cells form, you've got T cell precursors that leave the bone marrow. They get transported by circulating blood to the thymus. Once they're in the thymus, the T cell precursor becomes known as a thymocyte. So as the thymocytes slowly move through the parts of the thymus, the cells are going to undergo different changes. So 60 to 80% of circulating lymphs are T cells, which increases the likelihood of coming in contact with foreign antigens. This maturation process in the thymus takes up to about 3 weeks. Thymus secretions attract T cell precursors and cause that maturation. So the first stage of development is called the double negative stage, where you've got cytotoxic T cells and T helper cells that can be identified by their expression of cell markers, CD4 and CD8. And just kind of think about this as like the cell's holding up a little flag, and it says, this is my my identifying marker. So either CD4 or CD8. CD4 is used to identify T helper cells. CD8 is used to identify your cytotoxic T cells. But at this point, their fate has not yet been decided. So early thymocytes lack both CD4 and CD8. So that's why they're known as double negative thymocytes. During that double negative stage of development, rearrangement of the genes coding for those T cell receptors begins, and this is just a random process. It results in expression of unique antigen receptors by each thymocyte. So you've got a highly diverse population of T cells that are able to respond to a variety of foreign pathogens. Then next, that second stage of development is referred to as double positive. So at this point, the cells are going to express both CD4 and CD8, which is why it's called double positive. So the T cell must have a receptor for foreign antigen and self-antigen. So that antigen has to be presented to the T cells to test those newly formed receptors for its ability to interact through processes of positive selection. So again, we've got these we're forming these cells, and we want to make sure that they are performing and doing the job that they're supposed to be doing. where did I get to? So during positive selection, thymocytes that bind with very high affinity or those that do not bind at all are induced to die by apoptosis or cell death. So this means that the cell's not responding appropriately. It's either under responding or over responding, and we don't want that. We want the Goldilocks version. We want the one that's just right. So those that bind moderately survive, and they're going to proceed to the next stage of development. So after binding, they're going to express only either CD4 or CD8. So then the single positive stage, you've got self-antigens and peptide fragments that get presented to the positively selected thymocytes. So this stage is known as negative selection. So the reaction indicates high potential for auto reactivity, where your cells would attack your own body cells and tissues. We don't want that. So because of that, those cells also are selected to undergo apoptosis because they're not really under or over responding. They're just inappropriately responding. And then finally, our mature T cells. So once the T cell exits the thymus, it's considered mature. It's referred to as naive until they encounter their specific antigen. So cells are going to circulate between the bloodstream and lymphatics, waiting on that activation. So waiting to come in contact with their specific antigen so that they can become activated. Activated CD4, these are going to be helper or inducer cells, which will proliferate and secrete cytokines. And then your CD8, also known as cytotoxic or suppressor cells, respond to those cytokines that have been released. Then they proliferate and begin to destroy cells that display the antigens that activated that initial response. 2/3 of your peripheral T cells are CD4. Most of them are going to die in the thymus. Not that many of them actually get released. Next, your B cells are responsible for producing antibodies. So they're derived from a hematopoietic stem cell that develops into an early lymphocyte progenitor in the bone marrow and matures within the bone marrow. B cell precursors go through an ordered developmental process that prepares them for their role in antibody production and restricts the specific antigen to which any one B cell can respond. So just like with the T cells, they're going to go through this maturation process. Patients with B cell deficiencies will be more prone to bacterial infections. So with B cell differentiation, you've got the antigen independent phase. So this results in mature B cells that have not yet been exposed to the antigen. The pro-B cell, this is the earliest developmental stage derived from lymph precursor in the bone marrow for development. Cells receive signals that induce expression of transcription factors, which are proteins that control the expression of different genes. Then you've got the pre-B cell, which is signaled by the production of heavy chain mu, which is the first class of immunoglobulin is produced, is IgM. Your immature B cell, so this has a complete IgM molecule on its cell surface, the potential for antibody production to specific and unique epitopes. So the B cell that expresses a functional IgM B cell receptor does not react to self-antigens. And remember, that's what we want. And it begins to display certain markers. So at this point, it's considered mature. It will exit the bone marrow and it gets carried to the blood and spleen for the next stage. The mature B cell developed in the spleen. Both types produce antibodies. So you've got follicular B cells, which are constantly recirculating between the blood and the secondary lymphoid organs. They're on the hunt. They're looking for their specific antigens. And then the marginal zone B cells are going to remain in the spleen to respond quickly to bloodborne pathogens. B cell differentiation, you've got the antigen dependent phase. So B cells have been exposed to the antigen. So it usually involves the formation of plasma and memory cells. Also, your plasma cells are an abundant cytoplasmic immunoglobulin, but there's no surface immunoglobulin. The main function of plasma cells is antibody production. So this gets stimulated by that foreign antigen. They're found in the germinal centers of the lymphoid organs. They receive stimulation from cytokines from stromal cells, which are just kind of supportive cells. They're in the support cast in the lymphoid organs. Plasma cells are located in tissues. They're going to die after several days of antibody production. Plasma cells in the bone marrow are longer lived. The key surface marker on the plasma cells is CD138. Your memory cells, so these are long-lived cells that circulate in the blood. Natural killer cells, or NK cell, is the, I got behind, sorry. Here we go. Natural killer cells, third major population of lymphocytes. They're considered a type of innate lymphoid cell. So they have lymphoid morphology. They don't possess antigen-specific receptors. They don't have myeloid and dendritic cell markers. So they can kill target cells without prior exposure. So that includes the nonspecific killing of tumor cells. They mature in the bone marrow, not the thymus. They're found mainly in the liver, spleen, peripheral blood, where they make up about 10 to 15% of the circulating lymphoid pool. There are no surface markers that are unique to the natural killer cells, but they express specific combination of antigens used for identification. And then these are going to be the first line of defense against virally infected cells and tumor cells. Production of your natural killer cells are enhanced by cytokines such as interleukin and interferon, which are going, both of those cytokines are going to rise quickly during viral infections. At this point, it might be a good idea to pause this lecture video and watch the T cell and B cell supplemental YouTube video in Moodle. Okay, differentiation of T and B cells. So surface markers, which are cluster of differentiation markers, so those CD4, CD8 that we were talking about before, that's what that CD refers to, cluster of differentiation. So these are proteins on the cell surface that are used as markers to differentiate T and B cells. And it's also used to note, you can determine what developmental stage the cell is in. This is done through flow cytometry, and you may have talked about that process in hematology also, because that's how most hematology analyzers work, is through the a methodology of flow cytometry. So this is and you can see there's also a link to a video here. Flow cytometry is a system in which single cells or beads in a fluid suspension get passed through a laser light one at a time. So each time a cell passes in front of that laser beam, light gets scattered. So the interruption of that laser signal gets recorded. And that's done in two different ways, with both forward scatter and side scatter. So both are measured and analyzed in terms of their light scattering characteristics and intrinsic properties such as surface or cytoplasmic proteins. That gets done using fluorescent labeled antibodies or probes. So that process is referred to as gating. So your forward scatter is considered an indicator of size. The side scatter is an indicator of granularity or intracellular complexity of the cell. So those both of those measurements together characterize the different cell types based on their inherent properties. So those are what are referred to as intrinsic parameters, and then your extrinsic parameters require the addition of a fluorescent probe for detection. Fluorescent labeled antibodies are bound to the cell, and then they get detected with that laser. One of the big advantages of flow cytometry is that because the flow rate of cells within the cytometer is so fast, thousands of those cells can be analyzed in seconds, so it allows for accurate detection of cells that are present in very small numbers. Some of the more common applications for flow cytometry are characterization or differentiation of leukemia and lymphomas, identification of immunodeficiency diseases such as AIDS, detecting human leukocyte antigens or HLA antibodies in transplant patients, which we're going to talk about HLA more later. And then the major components of a flow cytometer include the fluidics system, the laser light source, the optics, and the photodetectors. And then you'll have a data analysis that occurs by a computer that's connected to it. And I thought I had a picture of a flow cytometer. There it is. So this is, you can see all the different, so you've got the cells that are going through the laser one at a time, and it's measuring forward and side scatter, and then all of this data gets sent back to the computer, and it'll give you a readout of those cell populations. Okay. Still talking about adaptive immunity, we're going to talk about the antibody response. So the primary response is mounted the first time an antibody is encountered. So it's characterized by a long time period, which is referred to as the lag phase between the encounter with the antigen and production of a detectable antibody. So that lag phase could be anywhere from 4 to 7 days. T and B lymphocytes are being activated to respond to the antigen by the T-dependent mechanism of antibody production. So this results in the generation of antibody-secreting plasma cells. Antigen-specific IgM antibody is produced first, then specific IgG antibody. The amounts of the antibody produced are relatively low. They're going to decline during the span of a few weeks. But because of this, because we have different types of antibodies that are produced during the process of a disease state or an infection, we can measure those IgM and IgG antibodies to determine what stage of the infection a patient is in. Okay, the secondary response, which is also known as the anamnestic response, you've got a shorter lag phase, usually about 1 to 2 days, so faster presentation of antibodies. You've got IgM followed by a greater predominance of IgG. So that patient would have initially a high IgG titer that declines slowly, but then persists for a long time. There's high affinity for the antibody to the antigen due to that recognition. So the body's already been exposed to the foreign antigen once. It starts producing those memory cells so that when it's introduced to that foreign antigen again, the antibodies that your body has produced have a high affinity for those antigens, those foreign antigens. Okay, acquired immunity is immunity that it's kind of what it sounds like. It's immunity that develops through your lifetime through exposure to various diseases, vaccines, etc. So you have acquired this immunity by being exposed. That comes in a couple different forms. So active immunity develops in response to infection or vaccination. And then you've got natural active and artificial active. So natural active immunity are antibodies that your body develops as a response to an actual infection. So you've been exposed to the actual organism or virus or whatever it is that causes that infection, and your body responds appropriately. It produces antibodies in response. Artificial active are your body's still doing what it's supposed to do. It's producing antibodies, but it's in response to an artificial source, such as a vaccine. Then we've got passive immunity, which develops after you receive antibodies from someone or somewhere. And again, we've got that can be divided up into natural and artificial. So natural passive are antibodies that are received from the mother, such as through breast milk. So this type of immunity is short-lived. So babies will have natural passive immunity or antibodies if they are breastfed. And then artificial passive, these are antibodies that are developed in response to an artificial source, such as globulin type medications or infusions that are injected. So this is an injection of pooled antibodies, not an attenuated vaccine that would cause the host to form an antibody response on their own. So these, I'm trying to think what it was called, during COVID, they used monoclonal antibody treatment. So what they did was they took antibodies from the serum of patients who had had COVID and who had formed those antibodies. They pulled them into a product that could be infused to a patient who was suffering from a COVID infection, and it only worked if it was infused early in the stages of the infection. And usually this is going to be for patients who are immune suppressed and are not able to form adequate antibody responses on their own. Okay, and I think, yeah, and here's just kind of a visual representation of that innate versus adaptive. And remember, innate is it's inherent in our bodies. It's those natural host defenses that are going to respond immediately and no matter what the pathogen is, versus adaptive, which is what it sounds like. Your immune system has adapted its response to that specific pathogen and can respond more specifically and faster. And that is the end of the first lecture for Unit 1.